I'd like to welcome everybody back. We're going to look today at photosynthesis and just a little bit of a reminder to you guys. It is about 6 o'clock in the morning right now when I'm doing this, so maybe a little bit forgetful at times. My speech may be slurred and I may fall asleep once or twice in the process of making this video. But I'm going to try to make it as exciting as possible because photosynthesis is a very important process. We are taking sunlight and carbon dioxide, we're essentially converting that to glucose and oxygen. So we've got a really intense process here because it makes all of the energy for the world. Think about it, the sun is the energy source for all life. And that sunlight has to get converted to chemical energy in some way, shape, or form. And that's the process of photosynthesis. So let's take a look at it. Today you will be able to give the formula for photosynthesis, analyze the organelles and materials involved with this process known as photosynthesis, and then understand the steps. Now, we're going to go a little bit more in depth than I normally would with my biology class. And the reason is not to scare you or to freak you out, but it's for you to understand and appreciate the complexity of this process. This is not just some chemical reaction that takes place. There's a long process that's involved with this, and there's a lot of different steps that are involved. And I just want you to be able to appreciate how complex this process is and how truly precious life is if you think about the complexity of this and cellular respiration. So the first thing I want to talk to you about is something that is called ATP, and that's short for adenosine triphosphate, and that provides the chemical energy for the cell. So any energy that is obtained is converted into ATP, and then when we utilize that energy, we take the ATP and break it down into ADP. So if you remember, when we make a chemical bond, we store the energy, and when we break a chemical bond, we use the energy. So when energy is stored, a phosphate is added to ADP, adenosine diphosphate, to make ATP triphosphate. So we're just adding a phosphate, as you see on the picture on the right. When energy is released or used, a phosphate is released or broken off of ATP. That chemical bond energy is released, and we have ADP plus energy that the cell can use for work or any other type of chemical synthesis. So ATP is essentially the currency, the energy currency of the cell. We make ATP in order to have energy, and then we break the ATP into ADP to use that energy. And that energy is used for almost all cellular processes. Just a quick overview of photosynthesis, and again, you hopefully have talked about this before, but we're going to take carbon dioxide and water and light, and those are our reactants to this chemical reaction. And we're going to produce a molecule of glucose, C6H12O6, and oxygen. All right, the coefficients are pretty important here, so I would make sure to know this chemical reaction, but it kind of makes sense the way you think about it. I always think about this as, what does a plant need to survive? Well, it needs sunlight, it needs water, and it also needs carbon dioxide. I always remember this is the opposite of what humans do. They take in carbon dioxide and exhale oxygen, whereas humans inhale oxygen and exhale carbon dioxide. So it's just an easy way for me to kind of remember how this works. And then we also get glucose out of it, which again, if you know, remember, it ends in ose, therefore it is a sugar. So there are two specific reactions that are involved here. There is what's called the light reaction, and then there's also what's called the dark reaction, also known as the Calvin cycle. Now, obviously the light reaction involves light. And we have these little organelles that are inside plant cells called chloroplasts, and these chloroplasts have the ability to absorb the light, except for green colored light, which is why plants tend to appear green. The chloroplast contains two compartments. The first is called the thylakoid, and that's the membrane inside that actually is able to absorb the light. So they're almost little tiny organelles inside of the organelle. And then outside of the thylakoid is what's called the stroma. A stack of thylakoids is known as a granum. That's just a stack of thylakoids that are really close together. So you have three vocabulary terms here: thylakoid, stroma, and granum. If we look at the thylakoid on a closer level, you'll notice that it looks an awful lot like a phospholipid bilayer. And you would be right, it is a phospholipid bilayer. But it has some very specific proteins and enzymes that are in it that allow for photosynthesis to take place. And you look at this diagram on the right here, it's very complex and confusing, but we're going to try to explain this as briefly as possible. So long story short, light causes water to split, and that produces some high energy electrons. And you see that in the first step there at photosystem 2. We have photons, which are little packets of light, and that breaks apart the water molecule, generating some electrons at photosystem 2, which is just a protein. Oxygen is produced as a byproduct of this chemical reaction, which lets you know where the oxygen comes from. The electrons then are transferred to a molecule known as plastoquinone. The plastoquinone then passes the electron to a proton or hydrogen ion pump known as the B6F complex. Now again, I understand it's a bit complex, but understand that the purpose of this B6F complex is to pump the protons into the thylakoid, creating a concentration gradient. We're pumping them into the thylakoid, creating a high amount of hydrogen ions on the inside and a low on the outside. So what this does is this creates a concentration gradient where the hydrogen ions are going to want to diffuse back into the stroma. We have a high concentration on the inside and a low concentration on the outside. And as you know, anytime you have something like passive transport, the hydrogen ions are going to want to go from high to low. And that's the purpose of the B6F complex. It uses those electrons to pump the hydrogen ions across the membrane into the thylakoid space. And then those protons are going to move back out of the thylakoid through a protein channel known as ATP synthase. And that's a protein that phosphorylates ADP to ATP. Essentially, it takes ADP and creates that chemical bond that makes ATP out of it. So this first process in the light reaction, we're really not even dealing with the breakdown of, or the creation of glucose yet. We're simply just making ATP so that we have energy to continue the process of photosynthesis. So in the light reactions, all we're doing is taking sunlight and water and converting that into ATP, which can be used in the dark or Calvin cycle. This process is known as photophosphorylation. Now that's a really, really long word, but break it down. Photo means light, and then phosphorylation is just the process of adding a phosphate to a molecule. So again, we're taking the electrons from water, transferring them through photosystem 2. Photosystem 1 takes those electrons and pumps hydrogen ions into the thylakoid space. Those hydrogen ions will then diffuse back into, or passively transport back into the stroma, thus generating ATP. Now we're going to use that ATP in the next chemical reaction. Now you may also look and see something that's called photosystem 1. And photosystem 1 absorbs light as well. Instead of passing it to plastoquinone, we're going to pass it to another molecule known as ferredoxin. And ferredoxin is going to generate this process where we take a molecule called NADP. NADP is actually short for nicotinamide adenine dinucleotide. All this is going to do is it's actually going to carry electrons. This is something that we can actually use to either use electrons from it or take electrons and use it to carry electrons to other parts of the cell. So at NADP reductase, which is another enzyme, we're going to take these electrons and convert NADP to NADPH. We're going to utilize those electrons later on in the Calvin cycle. So again, you see this complex process here. You've got the photons, and that's breaking apart the water, creating electrons at photosystem 2. Photosystem 2 transports those electrons to plastoquinone, and those end up going to the B6F complex. The B6F complex is then going to pump hydrogen ions into the thylakoid. The hydrogen ions are then going to passively transport back through the thylakoid into the stroma, and by doing so generates a small amount of ATP, which will be used in the dark reactions, or the Calvin cycle. So again, the dark reaction occurs in the stroma, and we're going to take the energy in ATP molecules that we created, and that's going to break down carbon dioxide into components that make up simple sugars like glucose. So we're taking the ATP and the NADPH that was created in the light reaction. We're going to break down carbon dioxide and convert it into glucose. It's very simple. We just use the energy, we use the electrons in NADPH to make glucose out of carbon dioxide. Now, CO2 is actually added to a 5-carbon molecule called RUBP. And RUBP is short for ribulose 1,5-bisphosphate. Now, I know it's a complicated term, but just know it as RUBP. So we take that carbon dioxide and add it to this 5-carbon molecule. Now, this makes a 6-carbon molecule. You think about it, 1 carbon from CO2 to 5 carbons makes 6. But this breaks down into two 3-carbon molecules called 3-phosphoglycerate. These are just precursors to glucose or sugar. So again, we're taking a 5-carbon molecule called RUBP, and we're adding CO2 to it, creating a 6-carbon molecule. But this is unstable and breaks down into 2 different 3-carbon molecules called 3-phosphoglycerate. Now, the ATP and NADPH that we obtain from the light reactions are used to convert this into glucose. Now, it's simplified here. It's a much more complicated process. But still, the concept is still there, where we're taking this these 3-carbon molecules, we end up combining them into something called glucose. So again, just a quick overview, and this is just very brief. We take the light. The light breaks apart water, creating some high-energy electrons, which help moves protons across the surface of the thylakoid membrane into the thylakoid. Those protons then go through the process of facilitated diffusion through ATP synthase to generate ATP, as well as creating NADPH. Those molecules are then transferred over to the dark cycle or the Calvin cycle. This takes carbon dioxide and converts it with RUBP to create 2 3-phosphoglycerate molecules. The ATP and NADPH are then used to generate glucose. So we've taken light, water, and carbon dioxide, and we have converted that into glucose, oxygen as a byproduct, and a little bit of extra water. I know it's a complicated process, but hopefully you'll be able to give me the formula for photosynthesis, analyze the organelles and materials involved with this process, and understand the steps of the process of photosynthesis. Sometimes it's just a process. That's all you really need to make sure you remember, that this is a complicated and complex process. Make sure you go through and understand how all these little components work, what they do, and whether they generate ATP, NADPH, whether they're generating glucose. Know the difference between light and dark cycles as well. All right, a little bit of intro to photosynthesis. We'll talk about respiration next time. Have a great day, guys. Talk to you later.